colleagues, for coming today to see my oral presentation about our recent paper. First, let me introduce myself to you. My name is Kunbaz. I'm from Syria. I'm currently first year medical student at Cerrahpaşa Faculty of Medicine, Istanbul University. Before that, I was student at Aydın Faculty of Medicine in Cairo, Egypt. And in this year, during my Turkish class preparation, I was working under the supervision of my dear professors Cakatay and Atukeren at the Department of Biochemistry. In this talk, I'm going to give you a brief introduction about our research, journal accepted, and its special features. Then, I'm going to talk about authors who contribute to our success. After that, I'm going to talk about our research, starting from introduction, going through methods, results, discussions, and then I conclude our results and tell you our future directions. After that, I would like to thank people and associations who supported us during our journey. Then, the last section will be feedback and questions. If you have any, I would like to answer it with the help of my professors. Now, as you see in the screen here, our research title is Oxidation, Scrutiny, and Persuaded Aging, Chronological Aging at Systemic Redox Homeostasis levels. This paper was accepted in one of the highest ranked journals related to biogerontology, which is called Experimental Gerontology. It's an American journal with an impact factor of 3.5. And let me say here that our paper was the first student project in the Department of Biochemistry, supported by TÜBİTAK Research Association in the Republic of Turkey and Istanbul University. Talking about authors who contribute to our success, starting from my dear friends in the fourth grade of Cerrahpaşa Faculty of Medicine, Tamer, Aydın, Tuna, Dr. Caroline, Associate Professor Pina, me, Dr. Mustafa, Dr. Riza, Professor Aydın, and Professor Cakatay, who was the corresponding author for this paper. Now, start talking about our research from the introduction. As many studies showed that D-galacose administrated animals showed the same characteristics physiologically and pathologically as their partner animals from the age group of 16 to 24 months. And as we all know, aging is considered as the highest risk factor related to many diseases and cancers. Unfortunately, every day, thousands of people are dying from diseases related to aging. And that's why aging process and its effect on the different body systems was studied extensively before by many researchers, including its effect on the systemic redox homeostasis. And it showed that the occurrence of the empiric redox homeostasis was associated mainly with the increase in the production of the active oxygen species. This increase leads to to increase in the oxidized lipid, protein, and DNA in the plasma, which lead to many changes in the endothelial cell of the arterial wall. And as we all know, these changes are favorable for initiation or progression of many superimposed vascular disease like atherosclerosis. Before, this idea thought to be apart from the normal aging occurring in all humans. But in our opinion, this idea should be reassured again after many studies occurred on D-galacose-induced mimetic aging. As the aging process and its effect on the different body systems was studied extensively before, and of course its effect on the systemic redox homeostasis, none of the study available in the current literature are talking about the effect of experimental aging on the systemic redox homeostasis. That's why the aim of our study was to clarify the effect of D-galacose administration on mimetic aging animals according to both its type and the increase in the oxidative damage occurring in its plasma. Talking about our research methods, before starting our research, all protocols were approved according to the national law of the Republic of Turkey and Istanbul University. Our study was occurred on 24 Sprague Dwali male rats. Those rats were housed in a temperature-controlled room with 12 dark light cycles, they were filled with full nutrients, and they had free access to water whenever they liked to. After the adaptation period, which was for 1 week, we divided them into 3 main groups. First group, 8 of them were mimetically aged rats with 5 months old chronologically. Second group was naturally aged rats with 24 months old. Last group was 8 of them with 5 months old, and this group is called young control rats. Now, regarding the mimetically aged rats, we injected them daily with D-galacose intraperitoneally according to this scale, 60 milligram for each kilogram of their body weight, and that lasted for 6 weeks. While as for naturally aged rats and young control rats, we injected them daily with sodium chloride for the same period. 24 hours after the last D-galactose injection, we sacrificed them under anesthesia. Then intracardiac blood samples were taken and drawn into tubes containing lithium heparin. After that, plasma were stored immediately at -80 degrees centigrade for the upcoming experiments. Now, let me show you here the oxidative stress-related parameters we investigated in our research. Starting from the protein oxidation biomarkers, we investigated the level of protein carbonyl groups, protein hydroxydes, advanced oxidation protein products, and protein thiols level in the plasma. Regarding to lipid deoxidation biomarker, we investigated the level of malondialdehyde, lipid hydroxydes, and conjugated ions in the plasma. Regarding to back oxidation adduct biomarker, we investigated the level of advanced glycation in products. Lastly, for antioxidant capacity biomarkers, we investigated the level of ferric-reducing antioxidants power level in the plasma, 2% superoxide dismutase activity in the plasma, and total and non-protein thiol fractions also in the plasma of these 3 groups. Also, we investigated the total protein content in the plasma according to biorate method. After all of these oxidative damage were obtained in the laboratory, we analyzed them, most of them spectrophotometrically and part of them spectrofluorometrically. Then, for statistical analysis and to compare our results, we used cross-cardinal biometric ANOVA test for comparing our results. Now, let's talk about our results in our current research. Starting from the first figure, talking about protein oxidation biomarkers in the plasma of these 3 groups. I'm going to use this abbreviation for shorted my sentence: YC for young control rats, NA for naturally aged rats, and NA for mimetically aged rats. First graph, talking about the level of protein carbonyl levels in the plasma of these 3 groups, we found that its level in mimetically aged rats were significantly higher in comparison to the 2 other groups, NA and YC rats. Also, we found slight significant increase in NA rats level in comparison to YC rats. Regarding to the protein hydroxydes level in the plasma, we found that its level in NA rats were significantly higher in comparison to both other groups, whereas no significant difference was observed between NA and YC rats. Then, for advanced oxidation protein products, we found that its level in NA rats were significantly higher, a slight significant increase in comparison to both other groups. Also, we found significant difference between its level in NA rats and YC rats. Last graph, talking about protein thiol levels in the plasma, we found that its level in NA rats were significantly lower in comparison to both other groups. Also, its level in NA rats were lower in comparison to young control rats. Second bar graph, talking about the lipid deoxidation biomarkers in the plasma, we found that regarding to malondialdehyde levels in the plasma, there were no significant difference among all of these 3 groups. For the second graph, talking about lipid hydroxydes levels in the plasma, we found that its level in MA rats were higher in comparison to both other groups, NA and YC rats, whereas no significant difference were observed between NA and YC rats. Regarding to conjugated ions, we found that the NA concentrations were higher in comparison to YC, lower in comparison to NA. Also, NA rats levels were higher in comparison to YC rats. Third figure, talking about glycoxidation other biomarkers in the plasma. We investigated the level of glycation end product and found that its level in MA rats were slight significant increase in comparison to both other groups. Also, NA rats level were also slight significant increase in comparison to YC rats. Last figure, talking about antioxidant capacity biomarkers in the plasma. Regarding to the first graph, talking about FRAB levels, we found that its level in MA rats were significantly and dramatically lower in comparison to both other groups, NA and YC rats. Also, in comparison between NA and YC rats, we found that there was no significant difference between them. Regarding to copper-zinc superoxide dismutase activity in the plasma, we found that there was no significant difference among all of these three groups. Regarding to total thiol proteins fractions in the plasma, we found that its level in MA rats significantly lower in comparison to both other groups. Also, its level in NA rats were significantly lower in comparison to young crows. For last graph, in the results section, regarding to non-protein thiol fractions in the plasma, we found that there was no significant difference among all of these three groups. Now, talking about discussions, due to its supremacy on the basis of systemic redox homeostasis, our paper could be considered as a fundamental study related to the systemic redox homeostasis. Chemicals, including D-galacose, are producing radicals, as we all know. These producing are lead to facilitate the aging process by induce many changes occurring in the different body systems, which is accompanied with impairment of the normal cellular function. Also, D-galacose administrated aging models offer many benefits to the researchers, such as easy application, short period for establishing aging model, and low cost. And we saw that D-galacose treated rodents showed similarity to the aging process occurring in the normal aging models. Many discussions were made between researchers about the optimal effect of D-galacose administration according to the dosage, age group, and time interval. That's why the selection of applied doses played a major role in establishing a successful aging model. Also, unapplicable doses used for the formation of mimetic aging model are affect mainly the redox homeostasis. Regarding to our results we saw before, as we all saw before, protein carbonyl levels were significantly increased in mimetically aged rats in comparison to the two other groups. Also, NA rats levels were slightly significantly higher in comparison to YC rats. While as for protein hydroperoxide level, we found that its level in MA rats were higher significantly in comparison to both other groups, whereas no significant difference were found between NA and YC rats. And due to protein carbonyl group stability in the plasma, regarding to the other oxidation biomarkers, we can say that it could be more reliable. Protein carbonyl group is more reliable and largely used marker than protein hydroperoxides for the determination of oxidative protein damage occurring in the plasma. Also, we found that the increase in the lipid hydroperoxide level found in MA rats could be considered as an accelerating factor for the propagation of other protein oxidation biomarkers such as protein carbonyl groups and advanced oxidation protein products, which, as we saw before, were increased in the plasma of MA rats. Now, the increase in the level of oxidation product seems to contribute to partial increase in other oxidation products such as AOPP, advanced oxidation protein products. As we all know, advanced oxidation protein products contain many other oxidation products such as protein carbonate, tyrosine, AGA, AGE, pentosidenate. Also, as we all know, conjugated dienes is considered as primary and early product of the lipid peroxidation. From this fact, and from the fact that aging process is a never-ending physical process, we conclude that it would be conjugated dienes level would be more prominent in acute incidence during sunsense. Now, our research regarding to antioxidant capacity, we found that the tendency of total fuel fractions and ferric-reducing antioxidants power levels in the plasma to decrease in both mimetically and naturally aged rats assure that our experimental models are applicable for the total CO status. Also, we found that the level of PSH protein fuel fractions were decreased sharply in mimetically aged rats comparing to the other two groups. From our results available in our research, it supports the argument about the D-galacose mechanism, about the action mechanism of D-galacose on experimental aging models. Now, let's conclude our results presented in our research. As we all know, natural aging process have limited aspects in research due to many factors like death risk, diseases, and high cost occurring in both humans and naturally aged experimental animals. That's why we conclude that natural aging process could be mimicked by D-galacose administration. Also, mimetic aging model may be considered as a reliable experimental aging model for the intravascular aging research. According to this point, in our recent paper accepted in the official journal of American Association for Aging, we conclude that mimetically aging rats could be considered as a reliable aging model regarding to myocardial sunsense. Now, to reduce the limiting factors regarding to D-galactose administration, it would be favorable to standardize the D-galactose model concerning its dosage and time period for upcoming studies. The last point, saying that oxidative biomarkers of aging need to be revisited again according to aging models after many studies occurred on D-galactose-induced mimetic aging. Talking about future directions, we think that additional methodological studies, including D-galactose administration, its dosage, and application time, are important to clarify its effect on all of these systemic redox variations. Also, the primary target for the development of efficient therapeutic strategies should focus on how to reverse or prevent the protein oxidative damage occurring in the plasma of both experimental and naturally aging animals. Now, after finishing our results and the section before the last one, I would like to thank TÜBİTAK Research Association and Istanbul University for their great support to our research. I would like also to thank my family, especially my mom, who supported me in all steps I took in my life, and of course for my dad, who dreamed to see me like a doctor. He did in Syria three years ago. I hope he's proud of me right now, and he's look at me right now. Also, I would like to thank my friends, professors, for their great support. And of course, I would like to thank you for your coming today. I hope you enjoyed this talk. Thank you so much.